The story of agriculture is the story of human survival. For millennia, we have shaped the natural world to feed ourselves. But nature, in its intricate dance, presents constant challenges. Disease, a relentless adversary, has plagued livestock since domestication began. Ancient texts detail devastating plagues that decimated herds, leaving communities in ruin. The spectre of such losses remains a constant threat in modern agriculture. Today, however, a revolutionary tool offers a potential solution: gene editing. Specifically, CRISPR-Cas9, a technology that has transformed the landscape of biological research. This precise molecular scalpel allows scientists to alter the DNA of living organisms with unprecedented accuracy. The question before us is profound. Should we harness this power to create disease-proof farm animals? The debate is complex, touching on scientific innovation, ethical considerations, and the very future of our food supply. The history of selective breeding provides a crucial backdrop. For centuries, farmers have chosen animals with desirable traits, gradually shaping livestock populations. This slow, incremental process, however, is dwarfed by the speed and precision of gene editing. CRISPR can introduce specific genetic changes in a single generation, bypassing decades of traditional breeding. The potential benefits are undeniable. Disease outbreaks can devastate livestock populations, leading to immense economic losses. Foot and mouth disease, for example, can cripple entire industries, impacting global trade. African swine fever, a deadly viral disease, has ravaged pig populations worldwide. These outbreaks not only affect farmers but also threaten food security. Gene editing offers a way to directly address these threats by introducing genetic resistance. Scientists have identified genes that confer resistance to specific diseases. By precisely altering these genes, animals can be made immune to pathogens. This approach differs significantly from traditional vaccines, which stimulate an immune response. Gene editing alters the animal's genetic code itself, creating a permanent resistance. The implications for animal welfare are significant. Reducing disease prevalence can improve the overall health and well-being of livestock. Fewer animals would suffer from debilitating illnesses. This could also lead to a reduction in the use of antibiotics in animal agriculture. Antibiotic resistance is a growing global health crisis. Overuse in livestock contributes to the emergence of resistant bacteria. Gene editing offers a potential pathway to reduce our reliance on these drugs. But the technology is not without its challenges. Off-target effects, unintended genetic changes, are a major concern. These unintended changes could have unforeseen consequences for animal health and the environment. Rigorous testing and careful validation are essential to minimize these risks. The ethical considerations are equally complex. Some argue that gene editing is an unnatural intervention, a violation of the animal's inherent nature. Concerns about unintended ecological consequences are also valid. Gene-edited animals could potentially interbreed with wild populations, introducing altered genes into the ecosystem. The long-term effects of such introductions are unknown. Public perception is a critical factor. Consumer acceptance of gene-edited food products is crucial for the technology's success. Transparency and open communication are essential to build public trust. Regulators face a daunting task in balancing innovation and safety. Clear guidelines and robust oversight are needed to ensure responsible use of gene editing. The cost of gene editing is another consideration. Currently, the technology is expensive, potentially limiting its accessibility to large-scale operations. This could exacerbate existing inequalities in the agricultural sector. Smaller farmers may not have the resources to adopt these technologies. The focus on disease resistance should not overshadow other important aspects of animal welfare. Good husbandry practices, proper nutrition, and adequate space are equally important. Gene editing is a tool, not a panacea. It must be integrated into a holistic approach to animal agriculture. The debate about gene editing in farm animals is not just about science, it's about our relationship with nature. It's about our responsibility to ensure a sustainable and ethical food system. It's about balancing the benefits of innovation with the potential risks. The future of agriculture may well depend on our ability to navigate these complex issues. Gene editing holds immense promise, but it must be approached with caution and foresight. The potential to eradicate devastating diseases and improve animal welfare is undeniable. However, we must proceed with humility, acknowledging the limits of our knowledge. The long-term consequences of altering the genetic code of entire populations are yet to be fully understood. The scientific community, policymakers, and the public must engage in a thoughtful and informed dialogue. This dialogue must consider the ethical, social, and environmental implications of gene editing. The goal is to develop a framework for responsible innovation that benefits both animals and humans. The challenge is to harness the power of gene editing while minimizing the potential risks. We must strive for a future where agriculture is both sustainable and ethical. The decision to use gene editing in farm animals is not one to be taken lightly. It demands careful consideration of all the factors involved. The path forward requires a balance between scientific progress and ethical responsibility. The future of our food system depends on it. We are at a critical juncture where scientific innovation meets ethical consideration. We must choose wisely, ensuring that our actions reflect our values and our commitment to a sustainable future. The power to reshape life itself comes with immense responsibility. Our choices today will shape the world of tomorrow.